Good afternoon, everybody, and welcome to the 2024 COVI Lecture Series. I'm Debbie Ingalls. I'm the Director of COVI, and pleased to see you all here today. It's my pleasure to introduce our guest speaker today, Dr. George van der Merwe, who's an Associate Professor at the University of Guelph. George received his PhD in microbiology from the University of Stellenbosch in South Africa. And during that time, he actually spent some time here at COVI during those PhD studies. He's a recognized expert in yeast nutrient responses and fermentation. He spent time during his career at the University of Stellenbosch, at the University of Tennessee in Memphis, Brock University here in COVI, and the University of British Columbia at the Wine Research Center. His own research program at U of G is focused on yeast in wine and beer industries, and I think we'll be hearing about a lot of that work here today. In the last 5 years, his group has focused on isolating, characterizing, and developing novel Saccharomyces and non-Saccharomyces yeasts for targeted application in the brewing, cider, and distilling industries. He investigates the responses and adaptations of industrial yeast to fermentation-related stresses at a genetic, molecular, and metabolic level. And he uses this information to develop targeted breeding pipelines to generate yeast with improved production efficiency and/or product quality and diversity in local fermentation industries. His research team employs standard microbiology, molecular biology, and cellular biology, as well as genetic techniques, and advanced approaches in genomics, proteomics, and metabolomics. George's research programs are and have been funded by the Natural Sciences and Engineering Research Council of Canada, Genome Canada, Ontario Genomics, Agricultural Adaptation Council, and the Ontario Ministry of Agriculture, Food, and Rural Affairs. That's a mouthful. So with that, please join me in welcoming George for a talk today in characterizing and developing yeast for the fermentation industry.
Thank you, Debbie. I came to COVI when that front part of the building didn't exist yet, and you were a postdoc in the lab with me. I did look for some pictures. I could thankfully not find any. that would have been different. But anyway, yeah, so it's been a while since I've actually been here. We've collaborated on some projects before. But I'm going to talk a little bit about the work that we've been doing with yeast, and mainly in the context of brewing. So a little bit more focus on beer, but the principles here apply to the wine industry as well, and also to the distilling industry, which is something that's of interest. So for me to give a little bit of an overview of what we expect the yeast to do, so this is old news for most of you guys, but we give it sugars and amino acids and some nutrients, and we ask the yeast to produce alcohol and all kinds of different metabolites, like glycerol and acetic acid, which is part of the central cost of metabolism. And that's pretty much what it does, and then it produces all these other kinds of compounds as well that gives it flavor. Either it contributes positively or negatively to the flavor, and we can measure these things. And consequently, we can measure the flavor compounds, and we can measure how it produces or uses sugars, and also alcohols, and ultimately at the end of the day, these readouts gives us an idea of its production efficiency, how well does it actually do the fermentation bit, and then also the flavor production at the end of the day, how it does so. This is our expectation of the yeast, right? That's not necessarily what yeast like to do, right? This is what we ask it to do. So if we think about this process per se, we break this down a little bit. You've got a variety of different kinds of sugars that the yeast uses, and there are all these kinds of transporters that bring these nutrients into the cell. And you've got fermentable sugars that the yeast can simply take up that are either mono or disaccharides, or trisaccharides for beer yeast that take these things up. And then there are also extracellular sugars that has to be broken down extracellularly and then consequently taken up by the yeast. So if we think about these kind of things, fructose and glucose are present in equimolar amount in wine yeast, in grape must. If you think about cider, there's a heck of a lot more fructose than there is glucose and sucrose. You think about beer, think about water, it's got very little fructose or glucose, but mainly maltose or maltotriose. Right? It's by far in higher concentrations than the other sugars. It also has extracellular sugars in the forms of complex sugar, dextrin, that it has to break down extracellularly. Many beer yeast can't do that. And consequently, these are taken up by transporters, and then there are a series of metabolic reactions that are performed by enzymes to produce these things, right? These metabolites. As soon as we put the yeast in a stressful environment, then that goes out the window, or it's impacted to varying different extents. And so this metabolism, these enzymes, these reactions we depend on, are influenced by stresses that we have within the fermentation itself. So what are these kind of stresses then that we have to consider when we think about fermentation? And so nutrients is the first one, is to be given enough of the nutrients that the yeast needs to perform the fermentation. That impacts how well it performs. There are inhibitory compounds that can be present within the substrate itself. Phenolics, for example, can be a compound that inhibits, right? Depending on the concentration and what kind of phenolics they are present. All right. A variety of different kinds of fungicides, for example, that are used in the field can influence how yeast perform. It's a fungus. Right. So consequently, there are a variety of different inhibitory compounds that can impact how the yeast perform, that are present within the substrate. Hyperosmotic stress is another one. Is how much, what is the osmotic stress or density of the substrate that's being fermented. The more sugar, for example, that's present within the substrate, the more osmotic stress there would be. So ice wine, in your instance, or high gravity worts, present a stress to the yeast that impacts that. Yeast like to actually ferment or like to do its fermentations at about 4.5 to 5 pH. That drops significantly during fermentation because it produces things like organic acids, and consequently that impacts how well the yeast ferments. The one that I want to talk about a little bit more in detail today is temperature. Is how does yeast experience temperature? We spend a lot of money on cooling fermentations down, or getting fermentations at a very specific temperature so that yeast can perform their function. Right. As soon as things start to warm up, then different kinds of structures within the yeast gets impacted. Proteins start to denature, the plasma membrane loses its permeability, or its permeability is impacted. The way that transporters in the membrane that I spoke of earlier, the way that they function gets impacted by temperature, and consequently, that gets impacted if we actually increase the temperature. The same thing goes for when we actually cool it down a lot. So in beer, there's lager production that takes place at like 10 to 12 degrees Celsius. And then you think about ice wine fermentation, that's typically in the, below 10 degrees Celsius is at least where it starts. So consequently, all those lower temperatures also impact how fermentation operates, how those transporters function, and importantly, how membranes function, not only the plasma membranes, but membranes within the cell. And so consequently, that's a big stressor, right, for yeast as they perform their functions. The other thing that we have to keep in mind is that there's also intracellular stress that we often forget about. So consequently, due to metabolism, there are a variety of different kinds of things that are produced within the cell that are inhibitory or even toxic. Reactive oxygen species is one of the main ones that's being produced during normal metabolism, whether it's fermentation or aerobic growth, doesn't matter. ROS is produced, and it influences, it oxidizes things like DNA membranes and proteins, and they lose their functionality. So we have to deal with that. There's also the production of medium-chain fatty acids as part of the fatty acid biosynthesis. So octanoate and decanoate, for example, are toxic to yeast, but they make them during fermentation. One of the ways in which they deal with that is to estrify them, make them less toxic. It doesn't mean that it's not, the fatty acids themselves are not produced. Ethanol by itself is a denaturing agent. Even though we love it when yeast make ethanol, they don't really love that a lot because it negatively influences them. Fortunately, Saccharomyces has an ability to combat that to varying extents, depending on what the yeast is. And then lastly, organic acids. They influence how metabolism is actually produced. Acetic acid is something that those of you that's come through Debbie's lab would be very well familiar with how acetic acid works and all kinds of other organic acids as well that lead to weak organic acid stress that impacts efficiency. So consequently, we have a variety of different kinds of stress conditions in which the yeast has to adapt to and to perform their fermentations, right? So that influences efficiency and also what kind of different metabolites are being produced and what the flavor profile of the yeast would look, of the product would look like at the end of the day. So how does the yeast cope with this? First of all, they have to obviously sense that this is around, so that these stresses are around, and then they adapt. There are a variety of different things that they can do to adjust to these stressful conditions. They can perform neutralizing reactions to take, for example, some of these stresses away. We spoke about esterification of fatty acids, that's one of them. Dealing with ROS, for example, would be neutralizing reactions. They can make intracellular protectants that will help them to combat these stresses. We'll get to some of them later on. They can alter their cell membranes. They can get rid of some of these toxic compounds by just simply pumping them out of the cell, or they produce chaperones, proteins that help to fold other proteins or protect membranes against these stresses. So there's a variety of different kinds of adaptive responses that the yeast can actually do to adjust to this. So what is it that I then do? What is it that we do in our lab? Is we try to identify all these stresses we spoke of before, characterize them in more depth, and we can talk about temperature a little bit. And then we use that knowledge to develop new strategies for the fermentation industry, in which we can either develop yeasts or strategies to do fermentations, in which we can circumvent or at least to some extent neutralize some of these stresses and give you a yeast that actually can perform its function more readily within the fermentation industry. Sometimes it leads simply to process efficiency that's better, or you can perform it potentially at a different temperature. Sometimes you also impact the diversity of the product. So consequently, you can make something that can make a product that has different kinds of flavor profile that's associated with it. But there's some variation that can come into that. I collaborate in my lab. I collaborate with a local yeast producer, Scotland Lab. It's out of shame plugging them here. And I work with them very closely. We identify industry-focused problems, and based on that, we develop research projects that we then carry forward. So stress responses and working with that, and how the yeast responds to nutrients are some of the things that have been ongoing now for a couple of years. We collaborate with some industries, most notably the cider and the fermentation or the brewing industry of late, to develop these products. So it's very much an industry-driven research program that I have going right now. And to get into a little bit of the genetics and the weeds a bit with looking at yeast. So we first have to think about domestication, right, in the context of fermentation. Many of the yeast that we work with have been used in fermentation for centuries. So consequently, they've adjusted. When you compare the genomes of a wild yeast to that of a yeast that's used in fermentation, they look vastly different. And consequently, you get, if you do that comparison, you get something that looks like it's a phylogenetic tree, or it is a phylogenetic tree, as indicated over here, that you compare to a standard yeast. And you can see there are a variety of different kinds of groups. This is the wine yeast, that's indicated over here. And then there are yeast with a mixed genetic background. We don't really know where all of these come from. And then we've got two groups of beer yeast. This one's beer 1, and it's got its origins in Belgium, Germany, US, and Britain. And then beer 2, that's up here. And they are genetically or phylogenetically very different from each other. Both the wine yeast and the beer yeast groupings, phylogenetic groupings, are very distinct, right? And they've been domesticated and being used in different kinds of fermentation, and that's why they are so different. During this domestication process, and I don't know if you guys can see that from the back, but for example, they've adjusted to use maltose for the beer industry and maltotriose. So the genes that's needed for that has been amplified, right? Within beer yeast, and you can see the beer 1 yeast here has, it's indicated in red, they've got multiples of these genes duplicated, whereas beer 2 does not. Actually, that's wine. It shows that for the wine yeast itself, it has not duplicated this specific maltose cassette for enabling maltose and maltose metabolism, because there's no maltose in wine. Right, it's glucose and fructose. Right? So beer yeast have been domesticated for that purpose. Beer yeast can also produce, or yeast can also produce phenolic off flavors. So they have the capacity to turn compounds, phenolic compounds that are present, like ferulic acid that's present within plant material, they can decarboxylate that to form 4-vinyl glycol, 4-VG, which is sort of spicy, clovey-like phenolic flavor associated with that. And depending on what the beer is or what the wine is, you don't necessarily like that to be present in the wine. So through domestication, through selection by the brewers, they've selected yeast that actually lost this capacity. So if you look, for example, at the beer yeast down here, this is a major group of ale yeast, beer 1. Most of them are blue, indicate beer, which means that they've lost the capacity to perform this reaction. However, there are some yeast in beer 2 that can perform this reaction, and they can make, for very specific applications, like Saison yeast, for example, that make that kind of beer, that have this clovey-like characteristic associated with it. Interestingly, some of the wine yeast used here are also positive for this, which may or may not contribute to something that the winemaker wants at the end of the day. I'll leave it at that. The other thing, so these are all traits that were selected for that contributes to the functionality of the yeast. On the other hand, they are also, through the degradation of the genome during domestication, it loses its ability to mate, to reproduce sexually. That's a problem, especially if you want to breed with the yeast. Right? So you've got to keep in mind that some of these yeasts have retained the capacity to actually perform sexual reproduction, whereas others do not. And it's important to recognize that some of the yeast, for example, in beer 1, have entirely lost this capacity that don't store it, and other associated functions, whereas in beer 2 it's variable. Wine yeast, interestingly enough, some of them actually retain that functionality, more so than beer yeast. So it's important to recognize this. So the question that we then have is, based on this domestication, we know that these yeasts have actually adapted to fermentation to a variety in different contexts. Can we use what we've learned through domestication of these yeasts if we look at their genetic makeup and we look at these adaptive responses that they have to fermentation? Can we combine those and actually come up with a yeast that's more tolerant to, for example, stresses like temperature, and then see if we can breed something that has newer application? The distilling industry can do with, for example, fermentations that are performed very readily without detriment at high temperatures. So every other industry, right? So the questions that we therefore ask is, can we identify and characterize the yeasts that have temperature-dependent traits that could diversify production? Can we make the fermentation to high temperatures or quality? How does temperature impact the yeast performance? And ultimately, can we unravel some of these traits by using high-throughput approaches like some kind of omics approaches, like genomics, proteomics, etc.? So we're going to touch on those things as we move forward. So this is where I want to introduce you to a group of yeast that you may or may not have heard of before. So there's this group, or this area in Western Norway, where they've been making traditional farmhouse ales for generations. We can date that back to pre the industrial age. The furthest we can go back is to like the 16th century, where they started making these yeasts in documentation that we have through historian that I'll introduce to you in a minute. So these little blue bulbs here represent different cities or areas where they've actually made, where they make this kind of product. So they make these traditional farmhouse ales. The way that they do this is they basically prepare wort, that is, and they let it cool down to the temperature that's approximately the temperature of a cow's udder, right? That's how it was listed in the historical references, right? So that we have since determined is around 35 to 37 degrees Celsius. And then they would seed that fermentation. They would add yeast into that. That's unheard of for what we currently do in our industry. We would never do that, right? Temperature is much lower because our yeast can't tolerate that. So how do they inoculate this? So they have this wooden ring, it's called the kwijk sokker. So these yeasts are called collectively kwijk. Right? That's the name for these Norwegian yeasts. So on this ring, they have a mixed population of microbes. That includes yeast and bacteria. They simply throw that in, fermentation starts, they yank it out, and they hang it up in the barn. Right? Now it's Norway, and so it's cold. Right? So these yeasts hang in the barn, it cools down significantly, it dries out, and then it's reused again, same population, to restart the next fermentation. So these yeasts are fermenting warm, and they dried and desiccated and cold adapted at the same time. So they're exposed to all these stresses in one fermentation round, and they've done this for 4 centuries, 4+ centuries actually. So consequently, these are super stress adapted organisms that's been through that for years. So what did they do? We expressed interest in these things. Richard actually heard about these yeasts in a conference where he was at. And so they shipped us a bag, subsequent bags as well, with shards of yeast that they broke off this. This is how it arrived at our lab, which is against every single protocol that you would hear of, right? It just arrived on our doorstep. We put these things in YPD because you've got to sweep this out. We put them in YPD. Richard went to grab lunch. When he came back, it was fermenting. That's the foam. Right? So they are super active. These beers are made within 3 days, and the fermentation is done. So what we did is we isolated these to purity, and we generated a strain bank. We've got about 300 of these strains, and we selected a subgroup of some of them, started to identify what they are like, what some of their characteristics. Did pilot fermentations in the work, and then we looked at their fermentation by measuring specific gravity, and we did HPLC to look at their sugar metabolism, ethanol production, and so forth, to get an idea of fermentation efficiency, and we did some GC analysis to look at their flavor compounds. We also, with a group of 6 of these, we sequenced them to see what their genomes look like. So the sequencing was done with short reads as well as long reads, so we could actually have a construct of what the entire genome looks like, not just SNPs, not only single nucleotide polymorphisms. So this is a group of people that actually started the work. So Richard was a master student when we started with this. Caroline Tiroir was also a master student, subsequently technician. And Christopher Krogeris is from VTT in Finland that we collaborate with, and this is our historian and expert on the history of kwijk and how it has progressed in Western Norway, Lars Hassel, and to this day we collaborate with all these people. We've got some papers that are in the pipeline as we speak. So what did we find when we looked at these things genetically? And so up here we have BR1 that I spoke of before, and down here is BR2, that's the wine yeast. And when we use the sequences and we actually look at the way it fits into the phylogenetic tree, it's a subgroup of BR1. But if you actually phase these genomes, you can see that you will see that we know that where one of these parents come from, the other one's unknown. We're currently in the process of developing a paper that will give us more insight into where that second parent comes from. This is some detail of the genome of the yeast itself. It is tetraploid. We've confirmed this with flow cytometry. And then here we also get some insight into its spore viability. As you can see that they have sacrificed some of the viability to some extent, where you've got around 50 to 60% for some of them, and then for some of them, like Storodol, for example, there's no viability that's associated with their spores. So it won't make. So that varies from strain to strain. You have to go test this. If we think of how these relate to each other in the principal component analysis, this is where the kwijk yeasts are. These are PR1s, and this is wine yeast. So they separate from each other quite distinctly through genetic analysis. They're not the same, but they are related. They show signs of domestication. I'm not going to go through the detail of this, but they're POF negative. For example, they don't have the capacity to form these phenolic off flavors. If you look at the genetic data here, this is the PAD1 gene, the first one, or the cofactor that functions in the decarboxylation reaction. It's performed by FTC1. These mutations are all high impact mutations. They are either premature stop codons or frame shift mutations. The numbers that you see here represent the 4 alleles that's present within the genome. 0 means no point mutation, 1 means a mutation. And so consequently, you can, for example, see for 1.1 that's homozygous for all these mutations. Consequently, it's lost its capacity to produce 4VG, which we've confirmed with GC as well. So not shown there. So that's the kind of genetic data that we've received through sequencing that confirms the phenotypic data. What's important to recognize here is that these point mutations also occur in most of the other yeast that are also POF negative. So through evolution, that happens all the time, that specific point mutation. It's a single point mutation, a single nucleotide change that alters the phenotype of this specific trait. The other thing that we just want to quickly emphasize here, if you look at maltose and maltodextrose metabolism, because these yeasts actually have to use high concentrations of maltose, when we look at the replication of different kinds of alleles or genes, this is a group of genes that are implied with maltose metabolism. There are copy number variations in the genomes of these yeasts, in which you have 14 copies, for example, in granulin 1 of the genes that's associated with maltose metabolism. Right? And so you've got these are copy number variations. So the exact gene is duplicated that many times to give you an idea of how efficient it can potentially perform its fermentation. So when we did a fermentation screen on these, we used a series of different quikes. So they are captured in the top part over here. And then our controls are down here, WLP001, or Cali, as you'll see later on. That's our control fermentation, and then the rest of them are all compared to that. And you can see some known beer yeast ferment faster than this commonly used one, but so does some of the quikes, which gives us an indication that these things ferment really efficiently. We look at flavor profiles. The gray boxes here indicate concentrations of flavor compounds that's produced above threshold. They're not only present, obviously, in quikes, but also in some of the control yeasts. But it means that they produce really interesting flavor components. In this instance, we're showing ethyl caproate, and decanoate that has fruity characteristics associated with it. So it has this capacity to make a fruity kind of beer. That's the bottom line to that. So they ferment really well. And that's the bottom line. Right? Is they can ferment, they make beer with an interesting flavor compound profile, but what else? When we actually subjected these yeast to different kinds of stresses for 24 hours and then incubated them in fresh media and to see whether or not they grew and just took optical density measurements, you'll see that most of these quikes down here improved upon what we would find in our control yeast that were down here. And so consequently, we came out with this is a temperature control yeast that's supposed to be temperature tolerant, which are the same or in some instances better than our yeast, the quikes, but it gives us an indication of how efficiently these yeasts can tolerate heat and ethanol. And so consequently, we came away with from our initial analysis that Richard did, we came away with the summary that this is a subpopulation of DR1, they're POF negative, they can metabolize maltodextrose potentially really effectively. There are some other traits associated with their fermentation that I didn't show here and that they're temperature tolerant. So the next question then would be, well, how well do they perform then if we actually change the temperatures on them? Because we're talking about higher temperature fermentations. That's what we want to do. So we have two people, David Foster, he's a PhD student in my lab, currently writing up his thesis, hoping to defend in 2 months. and then a bioinformatician, M&A, also in that actually worked with him. So he tested 4 quite yeasts. He tested, 6 of the quite yeasts, beer 1 yeast as controls, and then a beer 2 yeast as a control. And they wanted to ask the question, how well do they actually ferment at different temperatures? They went all the way from 12 to 42 degrees Celsius. And it took time points, as indicated over here, and we stopped after 5 days because we thought that the beers would be done by 3 days, 4 days. We started with an initial specific gravity of about 1.04. I believe that's about 12 or 13 bricks. Somewhere around there for context for you guys. So less sugar than you would have in one must, and that's our seed weight that we used. So first off, what's really interesting is the fermentation profiles that we have here in temperature. This is a loss of specific gravity, means that it's fermenting really well. The faster it can do that, the better it ferments. The color of the block indicates the temperature, and these are the controls at the top, beer 1s, beer 2, and then these are the quakes. And what is striking here is that the fermentation at higher degrees, at higher temperatures is absent, or there's no fermentation that happens in the beer 1s. And our temperature tolerance strain battles to do so at 42 degrees Celsius, whereas for some of these quakes, they really ferment really happily at these higher temperatures. So they can decrease the specific gravity really quickly. If we change the way that we actually portray that data, this is what it looks like. So this is after 3 days of fermentation. You can see that the line, the red line that's running through here is a specific gravity of 1.01, which is the target for ale production. You can see that the quakes at 35 degrees Celsius and at 37 degrees Celsius, 40 degrees Celsius, after 3 days they already reached that target. So the beers are done. They're made by that stage. If you think about how that happens for our controls, they tend to get there at 30 degrees Celsius, even though their fermentation is optimally at 22 in the industry. Right? And then after 5 days, you can see this is the fermentation profile. The interesting thing here is that our controls does not ferment at high temperatures. It doesn't reach, after 5 days, it doesn't reach our target temperature. It can do so very effectively at its optimal control, its optimal temperatures at 22 and 30 degrees Celsius, the green bars over here, but it can't do so at higher temperatures. And so it definitely is impeded to do so. So that's specific gravity. When we think about the sugar consumption, now I apologize for the busyness of these slides, but if we look at maltose, first off, the glucose and fructose concentrations are way too low to even mention here. That's all done within the first 6 to 12 hours. It's completely out of the picture. So maltose metabolism, then if we think about how fast these fermentations, how far their consumption goes, you can see this is temperature. At the bottom here, these are the individual, this is beer 1, beer 2, and quakes. If you look at how it consumes maltose after 5 days, it really battles to do so at higher temperatures, whereas at its optimum temperature, it has no problem. Quake's range of by which it consumes maltose is much broader than those of our controls, right? Even at the, especially at the higher temperatures, right? Now, the problem child when it comes to sugar consumption for wort is maltotriose. It's 3 sugars that are combined. Their consumption typically happens last when the yeast is the most stressed. And so one of the main problems with these spike yeasts rears its head here. Even at their optimal growth temperature, they don't consume all of the maltotriose. Right? If you compare that to the controls, they do a much better job, especially Kolsch, which is one of the beer 1 controls. It consumes most of the maltotriose at their optimal temperatures. However, the quakes never really do that. However, it can consume maltotriose up to above 75% at higher temperatures, especially for these 3 strains. So bottom line is that there's variability in how efficiently these quakes will actually use maltotriose, which means that you can't put them all under the same umbrella. They have really different phenotypes depending on what you're looking at. Right? Some of them are really more efficient at doing these kind of things than others. For example, Hornadel 1 in this instance has a much broader fermentation and efficiency profile than you would, for example, see for Laravel. Right? So you've got to pick the strain very carefully. You need to know what the strains can do before you actually use them. You can't bracket them all together. So the next question then is, well, how viable are these things then at different temperatures? So this is where the interesting bit comes in. We grew these strains at high temperatures, keep viability estimations of these 35 degrees Celsius. Our control strains were fairly viable. As soon as we got, the quakes are totally viable. As soon as we got to higher temperatures, there's some variation that crept in. The beer 1 control started losing viability quite drastically up to 42 degrees Celsius. Our temperature tolerant control was more tolerant than beer 1 as expected, the blue bars. But then when we look at the purple ones, you can clearly see there are some of them that are actually more viable than others. Again, there's a range of viability between these different yeasts, and some of them are super viable still at 42 degrees Celsius, which was a nice surprise to us when we actually saw that they can very happily perform their functionalities at that temperature. So then the question was, okay, well, why? Why is it that these things are temperature tolerant? So one of the adaptive responses that I mentioned to you before is the fact that it can perform or produce a protective compound that can protect its intercellular environment. And one of those things is trehalose that is present within the cell, and it responds to stresses that's being made in different kinds of stressful environments. It's a disaccharide, and it stabilizes membranes and helps to actually keep proteins functional. So when we looked at trehalose metabolism or production within these strains, at 30 degrees Celsius, the beer 1s produced some trehalose, beer 2 did not. And then there was a high amount of trehalose produced by our quakes. So it's like, okay, well, maybe we're onto something here. Maybe it's the trehalose that is actually giving us this protection. By the end of this experiment, a couple of days down the line, we still saw a high level of trehalose that's being produced and just decreased in the controls, which told us that once the trehalose is made, it doesn't really degrade that much when it's in the quake yeast. When we did the same experiment at 37 degrees Celsius, which obviously is a higher temperature, consequently we saw the beer 1 produces some trehalose and then it's gone after a couple of days of growth, whereas you still see this maintenance of high levels of trehalose within the quake strains. So what happens to trehalose is it's degraded by a neutral trehalase. It's an enzyme that breaks down trehalose. So we did trehalase activity assays on these yeasts, and our beer 1 controls have various efficiencies of trehalase activity, whereas our quake strains, for the most part, does not. So it lacks the beer 1 activity. When we went to our genome sequencing data, and I'm not showing you this here, but this neutral trehalase NTH1 has a couple of single nucleotide polymorphisms that's close to its active site that could impact its activity. We're currently busy in our lab to unravel whether or not that is the reason why we actually see very little trehalase activity, to link the genetic data we have to the phenotype. So trehalose activity production is one of the mechanisms in which quite can potentially result in its thermal tolerance. But we know that there are a variety of different kinds of stress responses that yeast can actually employ to combat temperature. So one of the ways that we can go about doing that is to do proteomics, in which we have a snapshot of the proteins done at different, of the different yeasts done at different time points during a temperature shift experiment. So Barrett did an experiment in which he compared a lab yeast that's very close to industrial yeast, a beer 1 yeast, and then a couple of quake yeast strains. And he grew that, he pre-cultured that to 30 degrees Celsius as a preheat stressor, preheat treatment. collected a sample, then he exposed this to a prolonged heat, a 6-hour heat treatment at 40 degrees Celsius, collected samples as a post-heat treatment, and then extracted the proteins and did proteomic analysis to look at what are the differences between these different strains. And so we first did some statistical analysis that could clearly tell you that the Vermont, the BR1s, is vastly different from our Quakes, and also, very surprisingly, our lab yeast control. That's very close. We knew it was very close to beer yeast. What's also interesting is that the pre-treatment is very different from the post-treatment in all instances. So there are significant shifts between the BR1 and BR2 as they respond to heat treatment. Not only are they different from each other, but there are differences within the treatment itself. So what are some of the interesting things that came out of this? We saw that central carbon metabolism, and I'm not showing all of this. There are significant changes. What we are comparing here is post-treatment to the pre-treatment, and everything that you see in red is upregulated, everything that you see in blue is downregulated. The first box is CNPK, is our lab yeast control, BR2 is the second box, and then the remaining 3 are Quakes. And everything that's in bold is statistically significant. So basically what you can see here is that all these storage carbohydrates, trehalose and glycogen, are impacted by the stress response. For us, it was really striking that you have trehalose, for example, that was impacted by a variety of these different yeast, of these different proteins that are misregulated. What we did know, though, is that this would have been a very confusing result for us, that the NTH1 is upregulated, in the sense that there's more of this being produced. Well, we know from our experiment that there's a high concentration of trehalose that's being produced, and we also know that this NTH1 is mutated from genetic information. So even though there's more protein, we know the protein's inactive. And so this is the importance of actually looking at what you see at all your data to intersect, and what does that really mean? Because if we didn't know the genetic data, we would have thought that there was more activity with NTH1, which is not the case. And so it's important for us to actually have a holistic view of how you actually interpret this data. So first of all then, we have a variety of different central carbon metabolism genes that are responding to this heat treatment, which gives us an idea of its metabolic activity. The other thing that's really important as well is that the way that it responds to ROS seems to be active. Remember, one of the intercellular stresses I spoke about before is reactive oxygen species. And that's the way that the yeast deals with that, is to perform neutralizing reactions in which it gets rid of the ROS, and there are a variety of different kinds of proteins that does that. And they are listed here, catalases, for example. You've got all kinds of thioredoxin, protein reductases, and peroxidases. And you see some of these are actually upregulated to a large extent within the Quake yeast, but not within Vermont. The second box is lowerly expressed, the second box is lower here and here as well. So consequently, this suggests that the Quakes actually have adapted a mechanism in which it can actually protect itself against ROS better than Vermont, for example, the BR1 yeast. And one of the things that I did when I came in this afternoon is I went onto my phone, and my lab technician emailed me is that he did the ROS screen and said that it works. Vermont has more ROS than Quake, so we now also have wet lab data to actually support this. In addition to that, the chaperone proteins that help to actually combat this seems to be upregulated, as indicated over here, and it's also higher expressed over here for to help with responding to those stresses. So what do we then do with all this information? Well, how do we go about and actually use this? So one of the things that I mentioned before is that we can harness that and actually breed yeasts that are stress tolerant and breed them with other yeasts. Maybe we can come up with something that's a little bit more resistant to stresses. So we did this with 2 yeasts. We did hybridization, which is not a foreign concept. I mean, you guys did this here at CCOVI a couple of years ago already. This is not new news, in which you can breed industrial strains, use 2 industrial strains, make them, and consequently have a progeny that could potentially have traits that come from both of those. We use the same kind of approach here, We use a quake yeast, which is one of these guys, or quake yeast that's in this grouping over here. It's phylogenetically very distinct from the beer 2 yeast. Remember, the beer 2 yeasts are the ones that have retained most of its mating capacity. The beer ones has lost most of those, so we're kind of limited with how efficiently that will work. It doesn't mean we're not still trying, we just haven't succeeded yet. It's a process. So we use 2 of these quake yeasts. We mated them to a beer 2 yeast, a saison yeast, which have very specific characteristics associated with flavor profiles. And we generated 2 strains, North Sea and Milner. And these are the guys that actually performed this work for us. Brian is my technician that is super capable and texted me the data on the RAS earlier today. So, first of all, let's look at the viability of these yeasts. So we did the same kind of viability assay we did before. Vermont is our control, it loses viability over time. The Belgian saison yeast, beer 1 itself, also has viability issues. It dies over a period of time at high temperatures, but it has an interesting profile for fermentation. And then these are our 2 quakes that maintain their viability. We have 2 different hybrids that are being produced, North Sea and Milner, from this, and consequently we see that they lose viability, or North Sea loses some viability fairly quickly, or it's fairly okay until 37, then it loses a lot of viability very quickly. Milner, on the other hand, seems to be viable for a longer period of time. That's an interesting strain, consequently, for us, because it can tolerate and efficiently ferment, hopefully, at high temperatures. So let's go ahead then and look at the fermentation profiles that we have associated with these. So here we have specific gravity decreases, again, with a target of 1.01. The blue line would indicate the saison, the blue line is 70 degrees Celsius, and the red line would be 37 degrees Celsius. So this is how they ferment. And consequently, the Belgian saison strain, it can ferment fine at 37 degrees Celsius, not a problem. The quake strains also get down to around that time period. We know that one of the quake strains has a problem at 22C. So consequently, this confirmed previous data. But when we actually looked at the hybrids, they both fermented really efficiently. If you look at the slopes of these, it's steeper than what we have with either of these parents, and consequently, they ferment really fast at 37 degrees Celsius. If we bar graph these, you can see that within 24 hours, Milner has actually already achieved its target specific gravity. So you can make a beer in 24 hours with this thing, right? If you let it go a little bit longer, and you can see a variety of some of these hybrids and the quakes actually meet the specific gravity requirements after 2 days, and then this is at the end of 5 days, where all the strains pretty much got to that. So it gives you an idea of fermentation efficiency and how fast these things can ferment. If we look at carbohydrate consumption, maltodextrose consumption, remember this is the problem child. We see that the maltodextrose consumption of the hybrids is lower than the parents in all these instances, so it can effectively utilize the maltodextrose more so than the parents. It's not at 0, granted, but it's very good for maltodextrose metabolism for yeast. If we think about dextrin, remember I mentioned to you that dextrin is an extracellular complex sugar that needs to be that can be degraded. That's not necessarily a trait that you want in all your beers. We call these beers the strains that can actually degrade dextrin. We call them diastatic yeasts. What they do is they excrete glucoamylase extracellularly that can break it down. That's great in beers where you want it. It's a tragedy in beers where you don't want it because it can lead to bottling problems. Right? So that can be a significant issue that depending on where you use it. Right? So that's a trait that has to be looked at after the fact. What we do see here, though, is that the parent strain, we know that the parent strain, the Belgian saison strain, this is a diastatic yeast, and consequently it would result in dextrin utilization. Quakes are not. They have limited capacity to actually perform that function, but we see that our hybrids inherited this trait from the parent, that it can actually break down dextrin, right? So it's acquired that trait. It has this diastatic trait associated with it. And then lastly, when you look at ethanol production, these kvikes or these hybrids produce ethanol basically the same as the Belgian strain. Same kind of context, right? So no problems there. If you look at acetate and glycerol production, so we know that glycerol production and acetate production are linked often through osmotic stress, in the sense that that the yeast produces glycerol as an intercellular metabolite to combat osmotic stress. In the process, it uses NADH, and then in an effort to recycle the NAD, it goes to acetate production in where it consumes NAD plus to generate NADH again. So when we look then at the production of glycerol and acetic acid in these different yeasts, we can see that the parent, the BR1, the BR2 parent, it produces a certain amount of glycerol that increases slightly, but not really very much. The stats isn't indicated here. So they come out in the wash. They're fairly similar between both strains, independent of the temperature that's being produced, where it's at. We see that one of these spikes actually produces more glycerol than the parent. When we use this quite in making a hybrid, the hybrids also have high amounts of glycerol that's being produced. By contrast, then, you also see that the acetic acid that's being produced is slightly lower than what you would expect, and we anticipate that there was this balancing reaction that took place for these, and consequently we have some indication of where we stand with acetic acid production and glycerol production. The limitation for sensory analysis is 200 milligrams per liter, I believe. So we're right at that with some of these hybrids. It's right there. So consequently, let's keep it in. I just want to leave some time for questions. So we've identified a variety of different kinds of cellular mechanisms that the yeast can actually use to combat increased thermal stress. These responses vary between these spike strains. And this is really important to recognize that you can't treat all of them the same. And that you can actually translate to any kind of yeast that you use within any kind of fermentation. You can't treat them all as the same because they're not. They're genetically very different from each other, and consequently that translates into how you can use them. Any kind of one-size-fits-all approach is likely going to experience some trouble in some challenge in one context or the other. Right? So it's a common way that people use to actually treat all yeast as if they are the same. That's not necessarily the best approach. But that means that you have to actually understand how these yeast function. You have to understand them, consequently you have to characterize them. The hybrids offer us an opportunity with selected hybrids to actually breed yeast that can have applications in different kinds of industries. And I'm not going to go into the detail of what these are, but typically the ones that are related to high temperature fermentations. So this leads to a variety of different kinds of breeding partners that could be available through these more stress-tolerant yeasts. With any kind of stress that we've thrown at these yeasts, they are typically more tolerant than most of the other yeasts that we try that we compare them to. We've only characterized 6 of them. There's hundreds more, and so consequently there's a lot of scope for development for a breeding context with applications in a variety of different industries. So the people that I want to thank would be our research team that I've spoken of throughout, our collaborators. These are the guys that gave us all the work to start these experiments with, and this is the people that pay the bills. So thank you very much.
We've got time for some questions.
Thank you for your talk. It seems that these yeast strains are very culturally important to this part of the world that still have a long history. Is this the first time that they've opened themselves up to research? Like, is this the first time?
No, it's actually gaining a lot of traction now. We were the first people to start publishing on this, but there are a few more papers that are coming out with different kinds of applications of these kinds of yeast with surprising substrates. Because they are so stress tolerant and they can actually withstand a lot of different kinds of environmental impacts, people are fermenting all kinds of weird things with it and consequently having success with it because they remain viable. One of the things that we do is that between Richard and myself, Richard more so, he actually goes back and he meets with these farmhouse farmhouse ale producers. They have a conference every year that he attends, and so he goes and he presents our research, and he speaks to them directly. And so we've got a collaboration going with them. And also, I don't think Lars is actually on this picture that I show here, but Lars Hassel, who's heavily involved with keeping the traditional brewing community together within Norway, and consequently we connect with him quite regularly, and also with Christopher Krogres. And as I mentioned before, we're currently in the process of actually delineating many of the yeasts in addition to these Kveik yeasts and what their origins are in a paper that we're hopefully going to submit by the end of March, beginning of April. So it's an active collaboration, and we work very closely with the Norwegian community to keep them abreast of what we're doing. They of course tell us that we can do better with ours because they have the community of yeast that work together. But we're working with individual strains, right? So there's a lot of scope for even developing that further. Yeah. They can be, yes. There are other ones that are also like this, that are fast fermenters, but that would certainly fall into that category, yeah.
George, I have a question for you. When you were characterizing these yeasts, you were looking at, their temperature tolerance for growth, and then looking at how fast they're fermenting. What about the aroma profile?
Yeah, so...
I know you probably have done measurement, there's a lot of data here, but yeah, I'm just interested on...
Yeah, so we did the aroma profile. I just mentioned briefly the aroma profile that we got originally. So we did aroma profile with the last hybrids, and then with the controls and the hybrids themselves. We got that data about 2 weeks ago, in which Brian processed the data and sent that to me, and we're starting to look at that in more detail. There's nothing that jumps out as terrible in the sense of, oh wow, it's producing this massive off flavor that we didn't expect. And we certainly didn't experience that during the production. However, there are some metabolites that are being produced that we have to do some thinking about explaining how that is coming about, thinking about how they are coming about. There are certainly differences. As you would expect, because they're higher temperature, you would expect that there would be massive changes, for example, in fusel oils and higher alcohols. There's not that big of a difference. That's different from the parents. We do see that there are, with these Kveiks in general, there are differences in fatty acid and fatty acid ester profiles. Right? And that's consequently where we see some of these differences coming. Right?
I was specifically going to ask you, what about volatile thiols? Because we know they form better at warmer temperature because the enzymatic reactions work more efficiently.
Yes, so if we can get a cheap way to analyze them, because if we want to ship them off to have them analyzed, elsewhere, you've got to sacrifice your eldest, your firstborn, to actually get that done. But no, it's for volatile thiols, we've used some of these Kveiks to actually, Escarpment has used some of these Kveiks to breed yeast that actually have a capacity to do biotransformations, especially of water precursors, that they would liberate thiols, right? Because they can actually perform that function. We know what the gene is that's associated with that. And consequently, we've got some information about that. We haven't done with our hybrids, we haven't done any volatile thiol work yet, mainly because we don't have the capacity to do that in-house at Guelph. But I mean,
We'll talk.
if we can talk, then that'll be great.
Yeah.
Yep.
Maybe a question. I know that you mentioned that when you collected these Norwegian yeast from different parts, one of the main characteristics is like they can withstand higher temperature, fermentation at this temperature. And then once you characterize all these strains, is there like any reason that you selected these particular 2 strains to work with?
Yeah. So first of all, you can breed with them. Right, so we test their spore viability and their ability to actually perform mating in mating experiments, and based on that, we have a short list that we actually pick from, and then we also look at what are the characteristics within those species, those specific strains. Temperature tolerance for the ones that we picked are the highest. Their viability is amongst the highest, and consequently, that's the reason why we picked them, is to go after those specifically. There are other yeasts, for example, that aren't necessarily as temperature tolerant, but they've got very interesting flavor profiles. Right, so that's subject to a different kind of breeding stream where you think about, okay, well, we're not looking at stress tolerance necessarily, but we want a certain flavor compound that we might be interested in pursuing. So it depends on what the trait is specifically. In this instance, we were going after temperature tolerance.
Well, George, a small token of our appreciation for you. Don't want you to forget about the wine.
Yeah, no. Thank you very much.
So
that's
please
awesome.
Join me in thanking George.
Thank you.
And then, just a reminder, please join us next week, Wednesday, March 13th at 2, as we welcome COVI's principal scientist, Dr. Sid Pujari, and he'll give a presentation on the importance of clean plant programs for grapevines. So we look forward to seeing everybody. Thanks.